So astrogliosis, this is really important because this is an inflammatory or an immune response to cell injury and cell death and cell inflammation in the central nervous system. Astrocytes are again a type of glial cell. So oligodendrocytes, good, they're a type of glial cell. Astrocytes are a type of glial cell, and they're activated in response to injury and cell death in the central nervous system. So in astrogliosis, you have an increase in the number of astrocytes, and when it's chronic, when you have a chronic increase in astrocytes, in astrogliosis, you get a formation of glial scars. And remember, we said in glial scars, we have these CPSG molecules that inhibit axonal repair. So if we can reduce astrogliosis, we can improve the central nervous system microenvironment, and we can hopefully improve the axonal regeneration. So just like I said, following trauma to the CNS, astrocytes can enter a defensive state known as reactive astrogliosis, which is characterized by significant changes in gene expression and function. And I said it's chronic. So chronic astrogliosis leads to the formation of glial scars. So in MS, astrogliosis contributes to the immune-mediated demyelination and neurodegeneration by increasing the amount of inflammatory immune cells in the CNS. It also results in blood barrier breakdown because we know that astrocytes, the foot of the cell of the astrocyte, binds and covers the tight junction of the capillary in the brain and the central nervous system. But when we have prolonged chronic astrogliosis, you have a breakdown of the morphology of the astrocyte, and it increases the blood-brain barrier permeability, and we get an influx of the pro-inflammatory cells. And in addition to the heightened immune cell levels, astrogliosis contributes to neurodegeneration through glutamate excitotoxicity. So glutamate is a neurotransmitter in the nervous system that excites nerves and excites cell. When we have too much, it causes toxicity and actually kills nerves in the central nervous system. So he goes on, the authors go on and talk about animal models of astrogliosis. So they state one study here. This study showed that 6 weeks of exercise decreased the expression of GFAP. Now, GFAP is a protein marker in astrocytes involved in CNS communication and function of the blood-brain barrier. So if we can have a decrease in expression of this GFAP, that indicates a decrease in astrocytes, and remember, astrogliosis is an increase in the number of astrocytes. So if we have a decrease in the number of astrocytes, we have a decreased amount of astrogliosis. So this study showed that 6 weeks of exercise decreased the expression of GFAP in the corpus callosum in MS model, in demyelinated model animals, suggesting a decrease in astrogliosis, and this was done in 2019. Now this is consistent with other work showing that exercise decreased astrogliosis and improved functional outcomes in other disease models. So for example, disease model of Alzheimer's disease, long-term treadmill exercise reduced astrogliosis and increased neuronal density. Now I'm going to put this up at the beginning of the video, but this last picture is a slide from the authors that summarizes the benefits of exercise in the central nervous system in MS model mice. So remember that they want to talk about oligodendrogenesis. So if we can increase the number of oligodendrocytes, that means we can increase the number of myelin sheath and the regeneration of myelin. If we can reduce pro-inflammatory and increase anti-inflammatory, we can improve the inflammatory environment and the chronic nature of inflammation in the central nervous system, and we can reduce the damage to the central nervous system because of this inflammation. Exercise also causes neuroprotection because it decreases demyelination. It decreases brain volume loss, especially in the areas relevant to cognition and memory, the hippocampus and the dentate gyrus. It also significantly increases the number of neurotropic factors such as BDNF, which are responsible for neuroprotection, neuroplasticity, creating new neurons, creating new axons, creating new blood vessels, etc. And we know that exercise, we've seen, especially in other animal models, it results in axonal regeneration and a reduction in astrogliosis. Now, this was a very technical article, and I hope I was able to explain the benefits of exercise in terms of CNS outcomes, central nervous system outcomes, without confusing anybody too much. My next article that I'll choose is going to be a lot shorter and focus on one point instead of several points. And please, again, if you have any suggestions for anything you want me to talk about, anything you're experiencing that you would like me to explain or find research for, just leave it in the comments below, and I'll do my best to attend to these and find research and talk about it in ways that you can understand. Please subscribe so you can keep up with the latest content, and until next time, thank you. Bye bye.